Hi everyone, welcome to another round of worldwide neural development. Today's main speaker is Klein, who will introduce you very shortly. But before that, we have the short talks, and guest today is Smith, Chris Lab at Harvard. And the floor is yours, Rich. Go ahead.
Thank you very much for the introduction. I'm very excited to tell you all a bit about some of the recent ongoing work in our lab, specifically studying ion channel genes and their role in developmental processes and neurodevelopmental diseases. So previously, we identified a voltage-gated sodium channel, SCN3A, and correlated mutations in this gene with malformations of cerebral cortical development. These malformations result in pathophysiology that looks like polymicrogyria, and so that is a fusing of the gyri and sulci in the developing brain. And then that's comparable to normal gyri and sulci here, where you get very clean white-gray boundaries. So we wanted to study a cell type basis of this particular disease, and one of the things that we identified from this study when looking at SCN3A was that not only was the voltage-gated sodium channel present in the cortical plate where the neurons are, but we also found a pretty abundant expression in the outer, in the radioglial, so the dividing progenitor cells. And this got us thinking more about the role or non-canonical roles of sodium flux or sodium in the developing brain. Moreover, we can perform, we wanted to identify exactly what this variant was doing to our voltage-gated sodium channels. And right here, to summarize, we perform electrophysiology. And what we find is that these mutations across the individuals increase the amount of sodium flux into the cell. So we have what we describe as a gain of function. Now, after we spent time working on this voltage-gated sodium channel, we were thinking, well, is this a voltage-gated sodium channel specific effect, or is it a sodium accumulation in the cell? So like I said, we have sodium that's coming into both progenitors and neurons. We accumulate in the cell. And it's the job of a protein here called ATP1A3 to pump sodium out of these cells. So we wanted to look closely at sodium-potassium ATPase, specifically the alpha-3. Like I said, this is present in neurons and it's very abundant in the brain. And we wanted to know if other genes, specifically ones that regulate sodium, are capable of also generating brain malformations. So when we look at our cohort, as well as with colleagues, we're able to identify ATP1A3 variants associated with polymicrogyria, very similar to our SCN3A individuals. When the cases we've seen so far are all de novo. The presentation is more heterogeneous than our SCN3A cases. And these individuals have developmental delay as well as seizures in the postnatal period. So it's a little bit different than our SCN3A mutations, which don't have that classic developmental delay or epilepsy. When we looked a little further into where these mutations are located on the actual protein topology, we see that they're actually restricted to a specific transmembrane 7 and 8 between these two domains in both the extracellular space and the intracellular space. And what this gets us thinking about is what exactly these mutations that are enriched to this area may be doing to the pump function in this particular subunit, this domain region. We know that has key interactions with beta subunit there, as well as other kinetic features. We can use human fetal tissue, so cortex, perform slicing and take a look at exactly where ATP1A3 is located in the cerebral cortex. And what we find is that it's enriched to the cortical plate area. So we see this enrichment, especially in deep layer neurons, but also we see some expression down here through the intermediate layers more sparsely, but there are levels of expression. We can take this same tissue and perform single cell sequencing. So we've generated a library over 125,000 single cell neurons. And the short story of it is that ATP1A3 is largely expressed through several populations of neurons. You see down here, EN stands for excitatory neuron, IN is inhibitory neuron, IP intermediate progenitor. And we see that there is levels of expression through various cell types. Obviously, no expression in astrocytes or cells. To go a step further, we moved forward a year in development, and we performed the same similar types of single cell expression analysis. And I just put it here as a summary slide to not overwhelm you with data. But what we found was that ATP1A3 was actually enriched largely to inhibitory neurons, and fast spiking inhibitory neurons. And this begins to let us think about this disease from a developmental perspective, from not only from the prenatal period in developing into these cortical malformations, but also the early postnatal period with our epilepsy phenotypes. The other thing that's worth noting is that we also notice the regionalization of this expression pattern, where the frontal areas of the brain in both the developing cortex prenatally and in the postnatal cortex seem to be enriched to this frontal region. So once again, thank you for your time. I hope I kept it under 5 minutes. And thank you to the many people involved and Chris for in the project.
Wonderful. Thank you, Richard. Ruediger, do you have a question for Richard, or should we go ahead?
No, I think we should go ahead.
Okay. One last shameless plug is that I'm also applying for jobs now. So if anyone sees a fit within their department, I'd really, appreciate any sort of consideration.
That's great.
Please feel free to reach out.
If you're interested in Richard's work, don't hesitate to send him a drop.
Yes, thank you again.
Okay, yeah. So I'll need you to unshare and then the. Okay, and for the featured event. So it's a pleasure to introduce Ruediger. Ruediger actually interviewed me on my first job application simultaneously in the same month. So, or at least in a few weeks of each other. So, so I had it at EMBA and at the Max Planck, and at the time Ruediger was moving from EL to the Max Planck. And so he was there at my job interview at EL, and he went there at my job interview at Planck as a moving director. And did end up getting a Planck offer and take it. That's a different story. So that's also an anecdote. So I met Ruediger in 2001 when he was doing labs. And you can, I guess, and from the intro, Ruediger, in fact, he did a PhD in and he then did post with Mariano Cid, and then EMBL as a group. And in 2001, he moved from EMBL in Heidelberg to Munich at Reed, where he became director, and within a year later, as a professor at the University of, and where he's still at. So I guess Ruediger's main issues can be summarized as how do cells talk to each other during wire the brain? What are the cell biological bases of aggregation, migration, how coordinate their actions and all of sort of brain development, brain wiring events through the surface model that they express and use to discuss with each other, and how is it that the molecules can mediate sometimes completely opposed effects in different contexts, and what is the biological base of all of that, and it's crucial to do. And seminal, special reference, fluids, many other of research families. I'll tell you all about this today. He's won many awards. He's really a major leader in cell biology, no doubt. And he's also really fun to hang out with. And perhaps more important than all of this, Ruediger is like a large number of very good science, a big fan of bioengineering. And of course, we share that passion. Not the goodest part. He's the scientist. I'm not. But we are definitely bioengineering, and I think that's a wonderful tribute for a person to have. So, without further ado, the floor is yours. Please share your screen and get started.
All right. Thank you very much, Bassam, for the kind introduction. I hope everybody can hear me well. Unfortunately, Bassem's introduction was sometimes a little bit interrupted. Let's see.
Yeah, sound on my end is not working well, so I'm sorry you couldn't hear me very well. I hope good enough to know that there's a great sign. And a Panic fan. All right, let's go.
Okay, do you see my my presentation?
Perfect. Yes.
Okay. So whenever there's a problem with sound or video, let me know. So I'm not talking to the void here. So I'm happy that many people are here. Unfortunately, I can't see you guys. It would have been much better to have you see you all live. Hopefully, we can do this again in the second half of next year when everybody is vaccinated. Okay, so I'm going to tell you about intercellular interactions during neural circuit development, and I'm going to start with this slide here, and with a statement that is probably the bottom line of much of what I'm going to be talking to you, that many of these guidance proteins that are expressed on the cell surface are multifunctional. They mediate repulsion, adhesion, or migration depending on context. And I'll give you the example of the ephrin and ephrin F system, which many of us have been studying for more than 20 years, almost 30 years, I must say. And the ephrins are well known for their repulsive activities. So in the slide here up on the left corner, you see a growth cone that expresses an F receptor and is confronted with a gradient of ephrin A's. And very often the growth cone would turn away from the highest source of ephrin. In contrast, on the slide on the right, on the upper right, you see a situation when this axon has arrived in the final target. And you would have a situation like this where an ephrin is on the presynaptic side and the corresponding F receptor, in this case a B-type F receptor, is expressed by the dendritic Filopodium. Then that high affinity binding of F-ephrin would result in some sort of an adhesive or attractive event, and that helps to form a synapse. This is very much work from Matthew Dalwa and a few other people. So it's quite clear that Fs and ephrins can do both repulsion as well as attraction. Then I've also added a couple of examples on the bottom of the slide. The example on the left side is from Rudi Winkelbauer's lab, who studied cell migration in the gastrulating frog embryo, where mesodermal cells migrate on ectodermal cells. And this migration is facilitated by alternation between attachment and detachment. And Fs and ephrins in this system provide the repulsive force, as you can see in that inside box. So you would have ephrin, B-type ephrins, the transmembrane ephrins, expressed by one cell, and the corresponding receptors on the other cell. And when the two cells meet, you get a repulsive event, so they can detach. And by some unknown adhesive force, probably adhesion molecules, then the next round of attachment starts. Now, interestingly, on the right here, there's an example that was actually published in the same year from Till Marquardt's lab, who studied the tracking of sensory axons along motor axons. And he found that the sensory axons express ephrin A's, while the motor axons express the corresponding FA receptors. And in this case, they actually do the opposite. They provide the attractive force for the sensory axons to be able to attach themselves to the motor axons. And there's some unknown repulsion force that provides the necessary force for detachment. So from these two examples, you can already see that there is quite a bit of multifunctionality in the F-efferent system. So the slides that I put together will not only focus on Fs and efferents, but I will also talk about a few other molecules. I will focus only on cell-cell interactions, direct cell-cell interactions. So I will leave out all of the secreted axon guidance molecules. I hope I can give you at least one example, not from my lab, but you will see how the signaling changes when a ligand receptor system promotes repulsion versus adhesion. And I will also get into the developmental processes that they control. So I'll begin with the shorter part will be on efferents and Fs. I will present you a work that we published last year on how Fs and efferents promote repulsion. It involves a process called trogocytosis, which is a mild version of phagocytosis, basically nibbling on your neighbor. And then I will switch to other families of another set of proteins, the flirts, the latrophilins, and the teneurins, and tell you about how they form bi- and tripartite protein complexes, about the developmental processes they mediate, and I will mostly focus on work from our lab on cell migration in cerebral cortex development. So again, here in the bottom of the slide, you see again the sort of domain architecture of the F receptors. There's a bunch of domains that are connected to one another. They form quite as a rigid ectodomain, which form these clusters. That's part of the activation mechanism of Fs and efferents. This is a picture from Elena Seradiki's lab work in 2010 when she was still a postdoc with Yvonne Jones. And here's a picture of flirts, teneurins, and latrophilins and how they interact, but I will get into that a bit later in my talk. So trogocytosis, this is something we've been working on for quite some time. We didn't call it trogocytosis. We basically called it transendocytosis because we noticed that during this process, Fs and efferents were actually taken up by the opposing cell. Here's a mini review on trogocytosis in general by these authors here. They were commenting on a paper that appeared in Nature Cell Biology in 2016 in C. elegans, where primordial germ cells and endodermal cells come in close contact, and this contact initiates a process whereby the primordial germ cell forms a lobe, which is then taken up by the endodermal cell, and then the membrane is pinched off, and the piece that was taken up is then degraded. And they were studying the signaling mechanisms that are required for this process, some of which have been known from phagocytosis, like SET10, which is the RAC G2Base, dynamin, but other proteins such as this protein LST4 were new. So here is what I summarized in one of my reviews in development in 2012, basically summarizing the work from Manuel Zimmer when he was still a student in the lab in the early 2000s. And he had observed that when an efferent B cell comes in contact with a cell that expresses an FB receptor, this contact formation and signaling between the efferents and Fs causes the FB cell to pinch off a piece of the efferent cell, like shown here, and basically engulf it. And in most cases, the efferent cell was also able to do this. So it's almost like bidirectional eating, nibbling at the opposing cell. And this is a way for the two cells to get rid of the molecular tether that is formed by the F-efferent complex, and to rapidly repel each other. And if you block this process, then the cells are much more sticky, and they could not get away so quickly. This seems to be a common process observed for the efferent Bs, for the transmembrane efferent Bs. And I should say that for the GPI anchored efferent As, it is more often the case that atom-type proteases are activated and cleave the active domains of either the efferent or the F receptor. So in work which was done by these two postdocs in the lab, Jinji Gong and Tom Gaetanos, who have since moved on, and which we published in JCB last year, we've actually characterized the downstream signaling events in the F-efferent cells and compared it to phagocytosis. Here's a picture of from C. elegans, where one cell, phagocytosis, is sort of a dead cell, and some of the genes and proteins that have been characterized to be required for this process. Here on the right side, you see the RAC GTPase, you see the activators of RAC GAFs, you see actin polymerization, which is required. Here on the left side, you see this protein here, GALP1, which it was the focus of this paper. It's an adapter protein that has a phosphotyrosine binding domain. It binds to phosphorylated receptors, and then by doing so, recruits a signaling pathway that then recruits dynamin. And dynamin, of course, you know, is important for vesicle, pinching vesicles off of membranes. So I don't want to show any of the primary data. If you're interested, it's in this paper and also in previous papers. We basically make the point that the miniature-sized trogocytosis that you see happening between an efferent cell and an F cell activates similar signaling pathways that are activated in a much more bigger scale when a cell engulfs a dead cell corpse. So it involves F receptors, the TM type of RAC GAFs, RAC itself, and then polymerization of actin to be able to sort of enwrap this lobe or piece of membrane, the recruitment of the GALP adapter protein and of dynamin, which then helps to pinch this vesicle off. So somehow these two processes are related mechanistically, and we thought that was quite interesting. This was also a collaboration with Rudi Winkelbauer in Toronto, who helped us providing some data to show that GALP is also required in vivo during gastrulation. Okay, before I move on from the Fs and efferents to other proteins, I wanted to point your interest, or to this study from Matthew Dalba, which I thought was a very nice study. And he showed that for a Filopodium, a dendritic Filopodium, to decide to either connect and make a synapse or to reject a presynaptic partner, the only thing you need is a change in the dynamics of F receptor signaling. So he found a very good correlation between a rejection event and a very fast rising of F receptor kinase activity on the one hand, and a very good correlation between making a synapse and recruiting, for example, VGLOT1, and a very slow rise, or much slower rise in FB kinase activity and a more sustained kinase activity. So basically providing an explanation for how Fs and efferents can do both. They can repel and they can also promote adhesion, simply based on the dynamics of F receptor kinase activity. Okay, so I'm going to leave the efferents and Fs now and switch over to proteins that we've been quite busy studying. The flirts first, and then later Teneurins and Latrophilins. This work was very much driven by Daniel Del Toro when he was a postdoc in the lab. He has now left and runs his own show in Barcelona. He collaborated with Tobias Ruff, a PhD student who is now at ETH in Zurich. And much of the work was also in collaboration with Elena Seradaiki, who provided a lot of the sort of structural insight in these proteins and protein-protein interactions. So the flirts are actually quite interesting. They are single transmembrane proteins, actually upside down here. So this is the membrane, this is the intracellular part. Is a fibronectin type 3 domain, and then there's this banana-shaped leucine-rich repeat domain. And so what Elena's lab did, she made a crystal consisting of one of the flirt leucine-rich repeat domains and at least a part of the ONC5 actor domain, and she showed that the two proteins contact each other at these points here, the side of the banana and the very first Ig domain of the ONC5 receptor. The ONC5, probably most of you know, is a neutrin receptor, and when activated, induces a repulsive signaling event. And the reason why Elena looked at this is because previously, in this paper here that appeared in Embo Journal, we had found that the flirts are actually ligands for ONC5, and this is one way for them to mediate repulsion in trans. So when a flirt cell engages with another cell that expresses the ONC5 receptor. And then we had shown in this paper that although we did not study this fully, there's some evidence that radially migrating cortical neurons that have the ONC5D receptor respond to FLIR2 in the cortical plate and actually slowed down by FLIR2 ONC5D interaction. Now, Elena also mapped the part of the leucine-rich domain of flirts with which they engage in homophilic interactions. So one flirt interacting with another flirt on an opposing cell. It's a different interface. So Elena was able to identify the exact amino acids that mediate this interaction, and we could make mutants of the flirts that would either fail to interact with other flirts or with the ONC5 receptor by putting, for example, a glycosylation site here in this blue patch or in the yellow patch. Now we know that flirt interaction with latrophilin is also via this yellow patch. So it's not only flirt-flirt, but also flirt-latrophilin interactions that are mediated by this surface. And I'm show you in the next couple of slides that this interaction site is quite important for regulating the migration of cortical neurons during development, and that this rather than regulating the radial axis, it seems to be regulating their lateral distribution. And that was published in this Neuron paper in 2014. Let me show you some of the data. So we had generated conditional knockouts of the FLIRT genes, and we also generated a LAC-Z knock-in allele, so we could easily see the FLIRT positive cells in the cortex as blue cells. We find that maybe a third or up to half of the cells that migrate into the cortical plate do express FLIRT3. And in a normal situation where they still have one functional copy of FLIRT3, they distribute quite nicely in the cortex, at least here in this lateral region. If, however, we use the nesting Cree driver and remove all of, so the last remaining functional copy of FLIRT3, the cells show a different migration pattern. They seem to be migrating in these columns. So we could easily see some areas where there are no blue cells. And Dani, who quantified this, could very easily see this fluctuation in staining intensity. And this was highly significant when he looked at many embryos. Now, intriguingly and very interestingly, you would see a very similar behavioral change of these neurons when you overexpress FLIRT3 using neutral electroporation at E15.5, and then looking at the brains at E18.5. Normally, in a control situation, the cells will be evenly distributed. But if they have too much FLIRT3, they would also migrate in these clusters. And this is clearly dependent on this FLIRT3 domain that with which they interact with one another in a homophilic fashion, and not due to an interaction with the UNC5 receptor. So we think that this is probably driven by too much adhesion mediated by FLIRT. And here it's harder because it's a loss of function experiment, but we speculate that similar to the examples of the efferents that I showed you in the first slide, perhaps here we are disturbing the fine balance between attractive forces and repulsive forces that these migrating neurons need to migrate, and we are losing some attraction, and perhaps we're gaining too much repulsion, and that's why perhaps they're being expelled out of these regions where we see no blue cells. Okay, so this is as far as we have taken this mechanistically. Dani's interest then went in a different direction. He noticed when he looked at the expression patterns of these proteins that another FLIRT protein, FLIRT1, there are a total of 3, I should say. FLIRT1 was always co-expressed with FLIRT3. So he thought maybe the phenotype might get stronger if he knocks them both out. So he generated a FLIRT1, FLIRT3 double knockout. In this case, FLIRT3 is removed conditionally using the nesting tree, and FLIRT1 is a full knockout. FLIRT1 single knockouts are viable, while the FLIRT3 full knockouts are embryonic lethal. So this is sort of a combination of FLIRT1 null, FLIRT3 conditional. He then looked again using the FLIRT3 LAC-Z allele at this funny clustering event. He noticed that that was even stronger. These are actually pictures from the same embryo going from rostral to more caudal sections. He also noticed that because of this uneven migration, that at the end, in the upper cortical plate, the distribution of the cells was uneven. There were regions where cells were very densely packed alternating with regions where there were fewer cells. And then interestingly, he noticed, and this was a serendipitous finding, that sometimes the cortex was actually folded. And we define this as a sulcus, basically, and not so much a gyrus. And you see in a moment why. Here's another example of an embryo stained with XGAL to look at the FLIR3 clustering event. You can see here the clusters, and in one of these clusters, you see the formation of a fairly deep fold. So this is at E15.5. This is the first time point when we start to see the folds. And this is the time point when we can see these clusters most clearly. A bit later in development, shortly before birth, the folds are more prominent. They come in different flavors. For example, in this brain here on the right hemisphere, you see this fold here. It's enlarged here. It's quite deep, but the basement membrane is still smooth. So if you were to look at this from the outside, you probably would have a hard time seeing that there is a fold. Here's a more stronger example of a fold here on the left side. You see these two deep fissures. In this case, the basement membrane actually follows the fold deep inside the cortex. We can see the Cajal-Retjes cells also going deep inside. So this is easy to see from the outside. The two examples I showed before, you can see folds on one, but not the other side. This is most often the case. And here, Dani summarized where the locations where we see the folds of something like 8 or so embryos. We see them most often on the left side in the lateral region, sometimes here on the right side in the more medial region. This may be, now we think that this is perhaps caused by the nesting Cree driver. We have not quite the same pattern when we use a different Cree driver, for example, EMX Cree. The folds seem to be also found in more medial positions, and actually not as frequent as with nesting Cree. So we're still looking at that. It might help us to pinpoint the mechanism of where these folds appear. Very intriguingly, if you look at all the literature and the induction of folds in the mouse, it always has to do with a change in the number of proliferating cells. Most often, an increase of progenitors and also subsequently more neurons in the cortex. However, when we do all these stainings and analyses, we could not see that there was a change in the number of proliferating cells. So we had to look for something else. And the something that we found was a change in the migration speed of these cortical neurons. So if Dani, again, takes E15.5 embryos, this is when when we start seeing the clusters, he divides the cortical plate in an upper and a lower cortical plate and determines, in a sort of an automatic counting system, he determines the number of blue cells in these two regions. Then in the control embryos, there is at this time point about a 50% distribution. So there are as many cells in the lower cortical plate as in the upper cortical plate. However, when he looks at a bunch of these FLIR1, FLIR3 double knockouts, he sees already more cells in the upper cortical plate, suggesting that the cells were actually migrating faster than in the controls. And a similar picture emerged when Tobias did time-lapse imaging. So he took these cortical slabs in culture, kept them alive for 24 hours or longer, and he could follow all the tracks of the migrating neurons, looked at all the physical parameters, migration speed, and so on. And the only thing that really stood out differently was the time the neurons spend at high speeds. So in this graph here, every row is a neuron, and I'm only showing you the high velocity segments. So when the neurons are actually migrating at top speeds, in both the control case and in the FLIRT-1-3 double knockout case, you can see here on the right side, there's more yellow. The tracks are longer and more numerous, and this is what we found. So they seem to be spending more time at higher speeds. So this and some computer modeling convinced us that something like this might be going on. So in a wild type embryonic cortex, the migrating FLIRT positive cells, they seem to be engaging once in a while in a FLIRT sort of adhesive manner, which the consequence is that they all migrate fairly homogeneously with a similar speed up to the cortical plate, and the cell density in the cortical plate is then also fairly homogeneous. In the absence of FLIRT-1 and FLIRT-3, the cells tend to form these clusters, and the resulting cell density here in the cortical plate is then also not even. You see areas where you have high cell density alternating with lower cell density, and we believe that this is the place where then perhaps we have the formation of these cortical fissures. And the reason, the fact that we do not see any change in the proliferation, so we don't see more neurons being produced in these brains, we think that we are mostly observing cortical sulci and not gyri. We also looked at, since we are working in mouse, but we want to learn something about human cortex folding, we looked at the levels of FLIRT-1 and FLIRT-3 in the human cortex, compared mouse data with human data, mostly from Wieland Hüttner's lab. We normalized the transcript levels using housekeeping genes like GAPDH, and it turns out that we humans have much lower levels in FLIRT-1 and FLIRT-3. I'm only showing the results for FLIRT-1 here. The same is true for FLIRT-3. So the mouse levels of FLIRT-1 and FLIRT-3 are way higher, in the order of perhaps 10 times more in neurons here, for example, or radioglia cells, or if you look at the layers in the cortical plate or subventricular cell. So what are we doing now in this with this question? When our paper came out, Jeff Meklis wrote a nice little news and views on this, and he proposed that there might actually be a two-stage model of cortex folding. First stage, you need to expand your progenitor pool to generate more neurons, and perhaps mixed populations of neurons and glia. And secondly, you need to change their migration dynamics or migration pattern. Perhaps you should see local acceleration of neuron clusters like we see in the mouse, which then would promote sulci. And so for the past year or two, I should say, it's a long process. We've been trying to test this two-stage hypothesis. And one way for us to test this is to combine our FLIRT double knockout model with other genetic models, for example, that show progenitor expansion. And so the hope is that if we have a brain, a mouse brain that produces many more neurons, which normally just would, excuse me, would normally just produce a thicker cortex, but a rather still a smooth cortex. If at the same time we would take away the FLIRT 1 and 3 genes, we would get something like this here on the right. It's of course a very optimistic view. And so I was hoping to actually share some data with you. This is ongoing, but I cannot reveal the model that we are most optimistic with. But I can tell you that it might be working. So we'll keep you posted over the course of the next year to what extent this is might be true. Okay, the final 15 minutes or so, I'm going to be talking about the friends of flirts, proteins that flirts are flirting with, so to say. And these are latrophilins and teneurins. So we have seen a tremendous flurry of papers over the past year or two, much driven by structural work from the Ruediger and the Arak labs that have looked at various interactions of these proteins. And this is quite fascinating data. I was really intrigued by the teneurin, for example. Teneurin proteins, these are very big proteins. They're actually upside down in the membrane, type 2 transmembrane proteins. So the C terminus is actually sticking out, the N terminus is intracellular. The teneurin ectodomain consists of this big barrel called the YD shell and some other protein domains that share homology to bacterial toxins. It's quite amazing. And they form dimers in the membrane. That's why I drew a dimer of 2 teneurins. And crystal structure work from Elena and cryo-EM work from Arak's lab has now shown that the teneurin, the barrel of the teneurins interact with the most N-terminal domain of latrophilin, the so-called lectin domain. And the second domain of the lectin, of the latrophilin ectodomain, called the olfactomedian domain, is the one that binds the flirts. And so I'll show you a picture on the next slide. These 3 proteins can actually form a tripartite protein complex. So what have we learned about what teneurins and latrophilins do? I cannot summarize the whole literature. There's actually quite a large body of data from the flies on teneurins. Teneurins are really evolutionary conserved. But Leach and Lu's lab has published a couple of nice papers recently on Teneurin-Latrophilin function in the hippocampus. So it turns out that proximal CA1 neurons, so there's a topographic connection between the CA1 and the subiculum. The proximal CA1 axons target the distal subiculum, while the distal CA1 axons target the proximal subiculum. All of these axons express teneurin-3, and the distal subiculum neurons also express teneurin-3. And he showed in a paper 2 or 3 years ago that the teneurin-3 homophilic interaction and probably attraction is what makes these proximal CA1 axons target the distal subiculum. And in very new work, which is on bioRxiv, the Luo lab shows that the reason why they are not targeting the proximal subiculum is because these neurons express latrophilin 2. And the repulsive interaction of Teneurin-3 and Latrophilin-2 keeps these axons out of the proximal subiculum and makes them target the distal subiculum. So this is some very nice work. It reminds me very much of Efrins and F. Then there's work from the Sutov lab, also in the hippocampus, where they show that the specificity of synapses on CA1 dendrites is also triggered by latrophilins and teneurins and even flirts. So the proximal dendrites of the CA1 neurons express latrophilin 3, and they form synapses with the shuffle collateral axons, while the distal end of the dendrites express latrophilin 2, and they are the target of the entorhinal cortex axons, the perforant pathway. And through a series of elegant genetics, they show that it's actually a tripartite complex of teneurins and flirts on the presynaptic side that engages latrophilins on the postsynaptic side through some probably adhesive complex that helps to form these synapses. Now, in my lab, in collaboration with Elena Seradaki, we showed that a very similar complex, different isoforms of the same protein families are expressed already earlier in development when cells migrate, and we could show that a similar complex regulates the migration of these neurons in what looks like a repulsive for the event. So again, remember that I started this talk by saying that these proteins are probably multifunctional. But we don't understand the signaling, the downstream signaling yet. This is still work to be figured out. Here's the sort of this amazing structure of the Teneurin ectodomain, consisting of this gigantic barrel here out of these beta sheet repeats. Here's a view from the top, the barrel here. There's some toxic domain sticking out on one side. The interaction with latrophilin is on the opposite side and with the lectin domain. The olfactor median domain also engages a little bit the, I believe, the Teneurin beta barrel, but is mostly free, and there's room for flirt binding. So it's possible for Elena to model the flirt 2 into this structure. So to study the mechanism of migration and axon growth, Dani made use of this nanofiber assay. He can buy these sort of prefabricated nanofibers, and he can put a cortical explant on them. What happens is you can coat these nanofibers with your protein of choice, and the cells that migrate out of these explants actually like these fibers very much. They behave a bit like as if this was a radioglial scaffold. So first, when they come out of the explant, they are multipolar, but with time, a few hours later, they become bipolar, and that's when they start migrating. They can also switch from one nanofiber to another one, similar to what they do in vivo, in real life. So Dani began by coating these nanofibers with latrophilin because he had seen that radioglial cells express latrophilin, and he put explants on this latrophilin-coated nanofibers, and he could see that in comparison to like control protein-coated nanofibers, these cells, the migration of these cells was actually significantly slowed down. And he then used basically structure-driven point mutants of latrophilin that no longer can interact, for example, with the flirts. We call these FL mutants. So this would be a flirt-deficient latrophilin. He could see a similar effect in slowing down the migration. He would then use a Teneurin-deficient latrophilin, and he would also see similar effect, only when he used a double mutant. So a latrophilin ectodomain, which can no longer engage with flirt or Teneurin, this effect was at least partially rescued or not seen. These are migrating cells. Interestingly, out of these explants, you could also see axons growing out. And these axons were not affected at all by latrophilins. So we can see that this repulsive signaling only happens in one compartment, in the cellular compartment and the little neurites, but not in the longer axons. We didn't know in the beginning whether this was repulsion or attraction, because you can slow down the migration also by increasing the attractive force, then they would basically get stuck, or by increasing the repulsive force. It turns out it's repulsion. So he then used stripe assays, the famous Friedrich-Bonhoeffer stripe assays. He confronted the explant with stripes of either the latrophilin mutant, the double point mutant in black, or the wild type latrophilin in red, and he did time lapse, and he could see that when the cells are coming out of the explant, they would grow these little neurites, and these little neurites would very much be repelled from the pink stripe. I hope you can see this. Here is a neuron that tries to grow a little neurite into the latrophilin stripe, and then it retracts. Axons that are coming out of these explants are not, they don't care about latrophilin. And so these are almost my last slides here. We then, of course, wanted to see whether there's any effect in vivo. We started by using gain-of-function experiments. So we overexpressed Teneurin in the migrating neurons. We were hoping that by expressing more Teneurin on his neurons, that we would disturb the sort of tripartite stoichiometry of the complex and disturb the signaling. And this was exactly what we could observe. Here's a control situation of embryos that were electroporated at E15.5 and harvested at E18.5. Dani divides the cortex into a lower, middle, or upper part, and he would then quantify numbers of neurons. If he overexpresses Teneurin, you could see that there aren't as many neurons in the upper cortical plate. You can see here, and also the middle part. And if he uses a point mutant of Teneurin, which has a mutation on the barrel that no longer allows interaction with latrophilin, then that effect was rescued, or not observed, I should say. So here's the quantification. Just look at the numbers of cells in the upper part of the cortical plate. In white, you have the fraction or the number of neurons here in the control situation. In red is when Teneurin is overexpressed, and in pink is the number of cells with the Teneurin mutant. This is gain of function, and we also have, then we also looked at some, Tobias looked at some time-lapse videos. He labeled all of the neurons with M-cherry and the Teneurin expressing ones in green, and then he could see that they would linger more often and longer before migrating up into the cortical plate. This is all quantified. Here in red is the position of the Teneurin positive cells versus in gray the control situation, and in the middle, in the yellow, is the Teneurin mutant. And finally, we've done a bunch of ways to do loss of function experiments. One was to knock down Teneurin-2 using two different oligos or shRNAs. And again, if you look at this picture here, lots of neurons migrating up to the most upper part of the cortical plate, and in the presence of the shRNA, there isn't as many neurons here. And this is the second one, again, similar picture. And here's the quantification. And so that allowed us to conclude that that system is this complex between Teneurins and Latrophilin and flirt is probably also important for cortical migration in vivo. So here is how we envisage the three proteins to engage, similar to what Tom Sweethoff showed in the synapses. We believe that flirts and Teneurins are expressed in one cell, and they are engaging a Latrophilin in an opposing cell. This could be a radial glia fiber, but it could also be another neuron because Latrophilin is not only expressed in radial glia, but also in migrating neurons. What is interesting, we think, is that we see a repulsive event when these three proteins come together, and this may be due to the fact that we're looking at signaling in the cell bodies and small neuroids and not in the axons. While of course synapse formation happens in the distal axon. So in summary, here's what I talked about: a short excursion into F-efferent signaling, where I showed you a summary of a story on drogocytosis, a process that resembles phagocytosis and basically allows the repulsive, the detachment of the cells to happen. Then I talked about flirt, adhesion molecules, and cortex folding. As I said, work in progress. I hope we're going to be making more progress here very soon. And then the last part was on flirts and Latrophilins and Teneurins and how they guide cortical neuron by repulsion. This is a relatively old picture this is in pre-corona times when we were still able to have fun together without face masks. Here's a list of people that are currently in my lab. Some of the important people are my outside collaborators, Elena Seradaiki. I didn't mention Victor Borrell, who helped us very much with the cortex folding phenotype. Rudi Winkelbauer, I did mention. Here's Dani Del Toro. Here's Tobias Ruf. Sarah was also part of the story, and Gönül. And here are the two people that worked on the drogocytosis. And with this, I'm done. I thank you all. I hope you're still there for your attention. And if there are questions, I'm happy to answer them.
Thank you, Ruediger. There certainly are questions. I have one, and I'm going to start. So, wasn't clear to me, and I'm sorry if I missed it, but in the folding experiment with the flirts and so on, you show preliminary data that maybe if you increase the number of progenitors, that maybe you get even more folding, etc. Two quick questions. The first is, in that case, does the basement membrane fold as well, or is it still only the cortical plate? And secondly, how do you envision it? So to what extent is this more folding or better folding because you're producing more neurons and it's the neuronal migration? Or is it now by producing more or perhaps different types of progenitors, perhaps maybe more basal progenitors, that now there's flirt-based interactions between the migrating neurons and these kinds of basal progenitors that change the developmental program of the cortex? So is it all based on neural migration, or are we getting into a different kind of cellular mechanism because we're generating more progenitors?
Yeah, these are all very good questions. I can only speculate. So the cases where we have seen that the folds are very deep, we do see that the basal membrane also folds in. In the case they are more shallow, the basal membrane stays smooth. What happens to now in a case where we increase the progenitors and the neurons? My hope is that we're going to see things that look more like gyri and sulci, okay, outgrowth and caving in, which we haven't seen so much. We haven't seen in the FLIRT, in the simple FLIRT double knockouts. That's the only thing I can say now, and we haven't really done a deep analysis of these brains. They're just coming in.
All right, so let's start with questions from our audience. The first is from Shuba Tole. Could you talk about how the gyri and sulci form more or less at similar places in given species? Multiple mechanisms likely interact? ?
Yeah, so I think if my memory is correct, the main sulci in the giant cephalic species, they are conserved, but they're little ones. They're more individual. I think every person has his or her own folding pattern. In mice, probably it's more random, and it may be that the cortex of a mouse is less patterned into regions, but this is something to be figured out. I would very much love to see more numbers and really look into cortical regions, and also look at neural activity in these regions, cross folds, things like that.
Okay, so Maeva Andriatsilavo is asking, thank you for your talk. Is FLIRT in complex with UNC5 able to interact with other FLIRT proteins at the same time? So is it mutually exclusive? Is UNC5 a way of preventing this? Could FLIRT UNC5 be due to an inhibition of FLIRT homophilic interactions? Yes. In that case, the repulsion would be due to a loss of adhesion.
I think anything is possible. Work that I didn't mention, again from Elena Saradaki's lab, she showed that FLIRT, when engaged with latrophilins, they can still interact with UNC5s. And they even form large complexes. So I wouldn't be surprised if UNC5 can fit into the tripartite FLIRT Teneurin-Latrophilin complex. That UNC5 in some situations gets recruited and perhaps helps to repel. But I would also not be surprised if FLIRT has other ways of signaling repulsion. For example, via its own cytoplasmic domain, by recruiting other types of proteins.
Mm.
Very, very, very little is known about what these proteins actually do in terms of signaling.
Mm. So if Sunches is asking, in the FLIRT 1-3 KO, you assume to have more repulsion pushing the KO neurons in streams. How would this affect migration speed? Would you have zones with traffic jams and zones of higher speed? Very cool question.
Yeah, I think the formation of these streams, as you call it, makes the neurons migrate faster. This is what we see in the knockouts. Whether all streams migrate faster, I don't know, but we do see the faster migration. Why they migrate faster, I don't really know. That's what we observe.
Okay, so
we're only looking at the FLIRT3 proteins, the FLIRT3 cells. We haven't looked at the FLIRT1 only cells or the other neurons that do not express the FLIRTs. This is only a large subset of the neurons, but not all of them.
So Arthur Kanya is asking, are there flirt 10-3 interactions?
No. As far as I know, there's no direct flirt to neuron interaction. It's only via latrophilin.
Eliza Moreno is asking, how would the flirt folding mechanism fit with other already described cues? So I guess the flirt-driven cortex folding mechanism, how would it fit with other already described cues like TRNP1? Do you think they are part of the same cascade of events, or is it convergence of different mechanisms? So many different ways to achieve the same result.
Yeah, so TRNP1 induced folding was more the production of more cells. They did not look at flirt expression in the TRNP1, but this is something that could be looked at. Perhaps some of the ways to produce more neurons would also affect flirt expression. But this hasn't, as far as I know, has not been really looked at.
Okay, so Fernando Alcina is asking, do soluble cues like reelin induce expression or downregulation of flirts, 10-3, etc. in migrating neurons that could modulate neuronal migration speed?
That's a good question. It has not been tested. The only thing we have looked at whether in the flirt double knockouts reelin was expressed normally, and we didn't see any sort of difference. We didn't see any change in reelin expression. But
I guess you didn't do it the other way around.
No.
Like looking at, okay. So Chaitanya Dutta is asking, how do you think increased velocity in the flirt 1-3 KO might be related to cortical folding? So is there a relationship? I guess that's similar to the previous question about the relationship of clustering and speed. So how might those be related?
We think that because they migrate in these streams, it may be that the cellular distribution in the cortical plate is no longer homogeneous. It's more like you have patches of cells in high density alternating with some other regions where there's fewer cells. This might be might then become easier to fold this region. But it's also true that we find the cells when they arrive in the cortical plate, they look more immature, and this may also be a way to help fold. So these are the two things that we see.
Right. So then our buddy Alain Chedotal has two very important questions, but I'm going to read the more interesting one first. Okay. The more interesting one is, do you think that Bayern will win the Champions League again?
I think they have a very good team and there's a good chance.
I'm serious. That question is actually there. I'm not making it up. Okay, his second sort of science related. I guess we have to deal with science sometimes. What can you do? Do you now have tools that would allow looking directly at flirt to neuron expression in the developing human cortex? Because I think you only showed RNA-seq data. So can you actually look at
antibodies with antibodies to look at?
I suppose antibodies in situs. I'm not sure to neuron expression, so it doesn't specify whether it's protein or RNA.
Yeah, unfortunately, I don't think we have developed any more good antibodies, if I remember, and we haven't pushed this to look at more evidence for flirt expression in other giant cephalic species. I think it would be nice to do. We haven't done it. It's all Danny's fault because he left the lab.
I also loved your answer to the Byron question. It was measured and very scientific, like based on data. It's like there's a good probability because they are not revealing the passionate side. Okay, so Shingo Nakazawa is asking, saying, "Thank you for a wonderful talk." I agree. I have two questions. One, if the number of cells is not so increased in the FLIRT 1-3 double knockout, is the cortical size similar? So you get folding, but I guess in the knockout, the size of the cortex, is it similar or not?
I think it is similar, yeah. The thickness of the cortex,
That makes sense.
I don't think there was any big change.
And then the second question, that's a really good one. Have you ever tested the cellular identity of mismigrating cells in the Teneurin-2 experiment? Is there effect on fate, on identity, I guess, temporal identity or spatial identity or whatever?
It's a good question, yeah. No, we have not. We have not. But it's something that should be done.
Okay.
Great.
Well, we got through all the questions, so I hope the people who asked them are happy about this. Thank you very much for your time, Ruediger. It was great to see you again. I really hope we get to see each other again in real life, at the Allianz Arena maybe, or at a conference. Thank you everyone for hanging on to this late hour, and see you again soon. And of course, don't forget to keep following the development series, all the other series that are exciting on this forum. And if you're a young person wishing to give the 5-minute talk before the main event, please don't forget to get in touch with the Worldwide Neuro email, and Denis will find the slots for you.
Thank you. Also, I see all the little notes and the happy faces. So thanks everyone for joining. I'm so sad that I cannot see you and hug you, but we'll have to do this again when we next summer when we're all clean.
Let's hope the vaccine is really coming.
Yes.
Yes. All right, everyone. All right, Ruediger.
Thanks very much. Bye-bye.
Thank you. Bye-bye.